The 2020 authorization of mRNA vaccines followed more than 30 years of global research into lipid particles and synthetic genetic code. At the center of that effort is messenger RNA. It serves as a temporary biological instruction manual that tells your cells how to build specific proteins. Inside a human cell, your permanent genetic code, your DNA, is locked away inside the nucleus. But mRNA operates exclusively outside of that vault, in the cellular fluid known as the cytoplasm. Because the nucleus acts as a strict physical barrier, the mRNA in a vaccine cannot cross into that vault. It never touches, interacts with, or alters your human DNA. This image shows the technical makeup of a lipid nanoparticle, or LNP. Bare mRNA is fragile and degrades quickly. To protect it, scientists encapsulate the mRNA within this microscopic delivery vehicle using neutral lipids, cholesterol, and PEG lipids to stabilize the particle. Once injected, these LNPs merge with the outer walls of your cells. As the lipid shell dissolves, it deposits the mRNA strand safely into the cytoplasm. Here, cellular machines called ribosomes take over. They clamp onto the mRNA and read the sequence like a piece of ticker tape. Using those instructions, the ribosomes physically assemble amino acids into a specific 3D shape, the spike protein found on the surface of the coronavirus. Once the protein is built, the instructions are no longer needed. The cell's natural cleanup enzymes approach the fragile mRNA strand and completely break it down. Within a short time, your cell has manufactured a specific viral component on its own. The temporary instructions dissolve into nothing, leaving absolutely no trace behind. With the spike protein assembled, the cell transports the protein through the cytoplasm and pushes it out through its outer membrane, leaving it exposed on the surface. Your immune system constantly patrols for foreign shapes. When it detects this unusual spike protein, it sounds a biological alarm, deploying specialized defense cells called lymphocytes. These lymphocytes study the protein and begin manufacturing custom-built, Y-shaped antibodies. These antibodies are perfectly contoured to fit and lock onto the exact geometry of that specific spike. Think about this step for a moment. If the body has just spent all this time perfectly contouring these antibodies to this specific shape, what do you think happens the next time the immune system encounters it? Through this process, the body has created highly specialized custom defenses in a controlled environment without ever having to face the live, dangerous virus. The answer to that question is a process called immunological memory. Long after the initial spike proteins are cleared away, those custom antibodies and the immune cells that make them remain on standby in your body. If the real SARS-CoV-2 virus ever enters your body, the pre-existing memory cells instantly recognize the viral spikes and swarm the virus with antibodies, neutralizing it before it can attach to your cells. This mechanism is built on a 3-step loop: deliver the code, manufacture the protein, and build the immunity. Because the loop is so reliable, the technology is highly versatile. By swapping out the genetic sequence inside the lipid nanoparticle, scientists can write new instructions. Instead of a coronavirus spike, this same process can target the flu, HIV, or individual cancer mutations. By safely handing our cells the blueprints to build their own medicine, researchers have created a foundational tool for the future of immunology.